The forms and functions of RNA are incredibly diverse, as we shall see in this presentation. In the first part, I'll talk about the processing of RNA and how RNA can actually help process other RNAs. Then I'll talk about RNA interference, an important process that exists in eukaryotic cells whereby RNAs, again, help to control the synthesis of other RNAs. And finally, I'll talk about genetic code considerations, which will include how transfer RNAs are best prepared for the process of translation. Most RNAs that exist in cells end up in a form different from what they started, and this process is called RNA processing. The processing of RNAs can involve a couple of things. It can involve, for example, chemical alteration of the bases that happens in some RNAs, like transfer RNA. It can also involve removal, deletion, and alteration of the bases within an RNA sequence, as we shall see. Now, enzymes called RNases participate in the processing of ribosomal and transfer RNAs in most cells. The ribosomal RNAs are usually made as larger molecules called pre-ribosomal RNAs, and these are processed in the nucleolus of eukaryotes. There are small RNAs called SNO RNAs that help in this processing, as I will describe. Now, transfer RNAs are also processed, but interestingly, transfer RNAs are made on the pre-ribosomal RNA, meaning that in some cases they're made with the ribosomal RNA, and the individual transfer RNAs are chopped out and then modified as appropriate for their function. The processing of transfer RNAs involves quite a few enzymes, but one of the enzymes is actually not an enzyme, meaning that one of the things that catalyzes a reaction is actually another RNA called a ribozyme. This ribozyme is known as RNase P, and as we will see, it helps to form the proper 5' end of a transfer RNA. Now, transfer RNAs, of course, are the RNA molecules that carry the amino acid to the ribosome for translation. They are a single strand of RNA that you can see in the figure in the upper right, having a 5' end and a 3' end. The 3' end is that part that sticks up a little bit further on the right on that image. That 3' end with the sticking up part is actually a specific sequence that ends in CCA, and that CCA is added by a polymerase after the RNA has been processed. Now, chemical modifications of ribosomal RNAs and transfer RNAs occur in prokaryotes and also in eukaryotes, and these chemical modifications are quite common. They will actually change what the structure of an individual nucleotide base is within these individual molecules. Now, you can see, for example, in this figure shown on the right, the conversion of uridine, which of course is a U within a transfer RNA, to a pseudouridine, as shown on the right. And this transformation has added another nitrogen to the ring, as you can see. Now, ribosomal RNA modifications, in addition to some of the general ones involving clipping out of sequences, are also helped along by some small nucleolar RNAs that are given the name SNO RNAs. So these small RNAs actually help to process ribosomal RNAs to their final form. Base alterations, meaning the actual editing and changing of a base sequence within an RNA, can also occur, and this happens in some eukaryotic messenger RNAs, particularly among the lower eukaryotes on the evolutionary ladder, but it also occurs in humans as well. Now, messenger RNAs in eukaryotes are very different than messenger RNAs in prokaryotes in comparing the ways in which they get processed. Messenger RNAs in prokaryotes get very little processing, and the reason that this happens is because translation in prokaryotes is occurring almost at the same time as the RNA is being made. And this is possible in prokaryotes because prokaryotes don't have a nucleus, and transcription and translation are occurring in the same place. In eukaryotes, transcription is occurring in the nucleus, and translation is occurring in the cytoplasm. So between the nucleus and the cytoplasm, there's a significant amount of alteration that can and does happen. Now, I've drawn on the screen here a representation of a eukaryotic messenger RNA, and it includes some of those modifications that I talked about. First of all, at the 5' end, we can see that the eukaryotic messenger RNAs have a structure that we call a cap. We don't see that structure of a cap in a prokaryotic messenger RNA. At the 3' end, or the other end of the messenger RNA, we see what's called a poly-A tail. The poly-A tail is the addition of a large number of adenine residues or adenosine residues to the 3' end of the eukaryotic messenger RNA. This polyadenylation appears to play a very important role in the stability of the messenger RNA and also improving the efficiency of translation of the messenger RNA as it interacts with the ribosome. The chemical modification at the 5' end to put the cap on is an interesting one. It involves the addition of a 7-methylguanosine to the 5' end of the messenger RNA, but that 7-methylguanosine is added in an unusual way. It's joined in a 5' to 5' linkage, as you can see in the image there. That 5' to 5' linkage is very unusual in biochemistry, and this is the only place that it occurs. And that cap also appears to play two functions. First, it protects the messenger RNA against degradation by nucleases. Second, the cap plays a very important role in helping to start the process of translation in the ribosome. Now, the long string of A's at the end is, as I said, probably there to help stabilize the messenger RNA and function in transport. There's a sequence within the messenger RNA as it's being made that goes AAU, AAA. And when that sequence is encountered, it is a signal to the cell to chop the RNA and begin adding the poly-A tail to the end of that. Now, in the middle of the messenger RNA, we're seeing the final messenger RNA right here. But the pre-messenger RNA that existed may well have had other sequences that were present that are not present in this final version. The removal of those other sequences from the internal parts of an RNA is called splicing. And splicing produces a product that has those internal parts removed and the final product shown as you see on the screen. Now, splicing is an important process, and I should emphasize that splicing occurs almost exclusively in eukaryotes. There's only a couple of genes known anywhere in the prokaryotic kingdom where splicing actually occurs. Splicing occurs on almost all types of RNA found in eukaryotic cells, but most commonly we find it in messenger RNA. It is found in some tRNAs and in some ribosomal RNAs, but most commonly we find it in messenger RNAs. Now, splicing, as I said, involves the removal of sequences internal to a pre-RNA, in this case, a pre-mRNA. This removal of sequences takes out sequences in the RNA that we call introns. So thinking back to the original DNA had a sequence that was transcribed, and that DNA sequence contained intron sequences. The introns in the RNA that's produced get removed. And so that's what's being depicted on the screen. We see the removal of the introns by a structure called the spliceosome. And the spliceosome, as we will see, is a complex of RNA and proteins that performs this function. The removal of the intron results in production of a molecule we call a lariat structure. And all that really means is that that intron had one end of itself bound to another part of itself to make a sort of a loop structure, as you see on the image, that has a form like that of a lariat. The spliceosome is a complex of both RNAs and protein. The RNAs are called snRNAs because they correspond to what we refer to as small nuclear RNAs. And when complex to proteins, we call them small nuclear ribonucleoproteins, or snRNPs. Some people call those SNRPs, although that's probably not exactly an accurate pronunciation. Now, this image depicts the sort of chemical way in which this process occurs. We can see, for example, the pre-messenger RNA at the top. It has an exon on the left and an exon on the right. Now, I need to tell you what those terms are. The exons are the parts that get joined in the splicing process. The intron is the part that gets removed. Now, it's very important that this process proceed precisely, because if the splicing is made one nucleotide or another in the wrong direction, then that will also have significant effects because it will affect the genetic code that is coming in the messenger RNA that's being translated. So this process must occur accurately if the protein that is going to be made from this messenger RNA is itself going to be made properly. So there are a couple things that we see in intron sequences, and you can see them labeled above that are common among almost all introns. At the 5' end of the intron sequence, we see a sequence of GU. That almost always occurs and probably helps to make sure that the orientation and the removal of the intron happens at the same place every time. At the 3' end of the intron, we see a sequence of AG, and that almost always also occurs within introns. Then about 30 nucleotides or so ahead of that AG, there's a sequence that is an A, as you can see here, and that A is usually adjacent to a sequence of pyrimidines that are not shown on this figure. Now, in this splicing process, what happens is the A that you see on the image makes a nucleophilic attack on the G at the 5' end of the intron. That nucleophilic attack results in the covalent bonding between the G and the A, as you can see here, and we've started to form the intron. The structure gets resolved when the joining of the exon on the left occurs with the exon on the right. That results in the cutting of the bond between the G on the right and the exon. That results in the removal of the lariat structure that you can see and the production of the spliced messenger RNA here. Now, common intron sequences help to orient and make this process possible, and without these common intron sequences, the cell would have no way of knowing where to make the cut. Now, one other thing I didn't note about the lariat structure is that it has an unusual bond in it. You recall from the structure of nucleic acids that the nucleotides are joined together in a 5' to 3' orientation, such that the 3' end of one base of one nucleotide is covalently bonded to the 5' structure of the next one. So that 5' 3' 5' 3' carries all the way through an RNA and a DNA. But this bond right here is unusual in that instead of having a 5' 3' bond, it has a 5' 2' bond. And that's because the 2' hydroxyl is available and it's what's making the nucleophilic attack that I mentioned. It's an unusual structure, but this is the one place in biology where it occurs. The excised intron comes out as a lariat, as I said, and that lariat can also be used for other things. In a few cases, the lariat itself actually contains another gene. That's rather unusual. Or the lariat can simply be degraded and the bases used inside of that structure. Now, this figure shows the joining of the snRNPs to the intron for making this process happen. You can see in this process that U1 is added first, U2 is added second, and then the complex of U4, U5, and U6 are added third. Now, we're not going to look up close and personal at that individual structure, but suffice it to say that there's a very precise and specific structure that's created that is known as the spliceosome. That spliceosome, some people have compared to ribosome in the sense that they both contain proteins and they both contain RNAs, and they both perform functions on an RNA. Well, this one isn't performing translation, but this one is now excising introns. The snRNPs and the snRNAs that they contain appear to play a very important role in aligning the A that's going to make the nucleophilic attack on the G in the right orientation. So once that orientation is set up, then that excision of the intron and the formation of the lariat structure can actually occur. So there's actually a sequential series of events that happen in this process. After the structure has formed, we see removal of a couple of the snRNPs, the U4 and the U1, for example, and then the release of the others, and finally the spliced messenger RNA along with the lariat structure. Now, before I go further and talk about processing of other RNAs, I want to stop for a moment and talk about another function of RNAs because they'll relate to the processing of the transfer RNAs that I'll talk about. And these are the RNAs that are catalytic in nature. These RNAs actually catalyze chemical reactions. They're the RNA equivalent of enzymes. We call them ribozymes because they catalyze reactions. Now, ribozymes play roles in ribosomes, and I bring that up here because it's pretty easy to confuse those two terms. A ribozyme catalyzes a reaction. A ribosome is a big structure that makes peptide bonds in proteins. Ribosomes contain ribozymes. Okay. So, We can see that in the figure that's shown here. On the right, we see a ribosome, and we've seen this figure in another structure, in another presentation that I have made. In this reaction, what's happening is individual amino acids are being joined, as we can see here, in formation of peptide bonds. And during the formation of those peptide bonds, one of the ribosomal RNAs, specifically the 23S ribosomal RNA, in the case of a prokaryotic system, is making the bond between the adjacent amino acids. So that peptide bond formation is happening as a result of action of a ribozyme within a ribosome. Okay. So there are other ribozymes that play roles in processing of individual transfer RNAs. So let me show you one of these. This is a ribozyme known as RNase P. RNase P plays an important function in the final processing that's done on a transfer RNA. Now, it's pretty simple, but nonetheless, it's a catalysis that occurs. On the screen, you can see the transfer RNA, and at the 5' end, you can see a black line sort of looped off on the side. That 5' end has to be processed. It has to have that little piece removed, because without the removal, the transfer RNA will not function. That piece is removed by RNase P. So in addition to the many functions that RNAs perform in the process of translation, as you can see, they also perform functions of catalysis, and in some cases, those catalytic functions are on other RNAs, an amazing group of molecules. Now, that structure removed by RNase P is simply lost. It's not very long, but it's long enough that it actually would stop the translation process from happening if it were not removed. Now, another important process recently discovered that all RNAs also function in is a very critical and interesting process called RNA interference. Now, RNA interference is a process that's stimulated by the presence of double-stranded RNA inside of cells. Now, as we're going to see, this can happen either as a result of the cell making double-stranded RNA or by a virus that has invaded. And the invasion of a virus bringing in double-stranded RNA is a sure cue for a signal for a problem. Now, the two different forms of double-stranded RNA that can exist in a cell are known as microRNAs, or miRNAs, and these have cellular origins. The siRNAs, or silencing RNAs, come from an external source, like a virus, for example. Now, I note that biotechnologists are also using double-stranded RNAs as a means of controlling genes for biotechnology purposes. So that's another way that foreign siRNA could get into cells. Now, the process is quite widespread in eukaryotic cells. It occurs in plants and it occurs in animals, and it plays a wide variety of roles. The actions performed in this process are called RNA interference, or RNAi. And what these double-stranded RNA molecules result in is the interfering of the translation of targeted genes. Now, the ability to use this technology to target and specifically stop the function or stop the production of certain proteins allows a researcher to do incredible things, but more importantly, it allows the cell that has this to both protect itself from invaders and also to control its own gene expression. RNA interference operates through the silencing of gene expression. And that silencing, as I said, is an interference in the way that proteins are made. Now, this occurs as a result of, first of all, the appearance in the cell of a double-stranded RNA. And there's an enzyme called dicer. And just like a dicer that you might have in the kitchen, this dicer's job is to take that double-stranded RNA and chop it into bite-sized chunks. And those bite-sized chunks are about 20 base pairs long, as we shall see. So these 20 base pair chunks at this point are called silencing RNAs if they came from an external source, and microRNAs if they came in from a cellular source. These pieces of RNA can then be bound by the RNA-induced silencing complex, or what's known as RISC. Now I'm going to show you this whole process in just a minute. Now this figure illustrates the process that I was describing in words on the previous slide. We can see actually two things occurring right here. In the process occurring on the right, where I'm describing a cellular process that starts with the production of an RNA that makes a double-stranded RNA used to create the miRNAs that I described. In the process on the left, we have simply the double-stranded RNA that has appeared in the cell by the foreign source, whether it was a virus or it was by a researcher placing that within there. I'm going to start with the process on the right to describe it. The cell has encoded within its genome certain sequences that, when transcribed, produce structures like you can see on the right. That double-stranded structure with tails hanging off of it and the poly A that you see on it is called a miRNA. The name doesn't really matter. But that miRNA gets processed to make what will ultimately become the miRNA. So there's an enzyme called DRASA that cuts some of the ends off of the mRNA and creates the smaller structure that you see on the image. The mRNA is then moved out of the nucleus, as you can see, where it is then attached to the enzyme known as dicer. Now at this point, the two processes, the siRNA and the miRNA, become the same. What dicer does is it takes that double-stranded miRNA, or the double-stranded RNA that you see on the left from the foreign source, and chops them into the 20 base pair sequences that I described. You can see, for example, a perfect duplex on the siRNA side and sort of a mismatched duplex with a bulge on the miRNA side. That's very common for miRNAs. Dicer, after chopping this into 20 nucleotide blocks, then peels away one of the strands. And the peeling away of that one strand leaves a single-stranded siRNA or a single-stranded miRNA that is then grabbed by the RISC. Now the RISC takes that individual sequence and carries it to a messenger RNA. Now the significance of the fact that there's a single strand at this point is due to the fact that this single strand will be complementary to a target messenger RNA, as we shall see. Now, after the RISC has complexed with that single-stranded RNA, whether it was an miRNA or whether it was an siRNA at this point doesn't matter. That RISC RNA complex then goes and seeks messenger RNAs. Messenger RNAs, of course, are coding for individual proteins. If RISC finds a sequence that's complementary to the RNA that it's carrying, it aligns that sequence with the specific region in the messenger RNA, as you can see here, and then an enzymatic activity in the RISC complex called argonaute actually cleaves the target messenger RNA, as you can see here. Now that cleaving of the target messenger RNA means that you've destroyed the coding for a protein. In this way, this protein that was coded by this messenger RNA can no longer be made. Well, this has a couple implications, as you could imagine. This has very obvious protection effects for the cell against an invading virus. If the invading virus makes a double-stranded RNA in the process of its life cycle, then this siRNA system will stop the production of targeted virus proteins. Now, also, it may seem a little odd, but the miRNA also plays a role in regulating gene expression, because the miRNA is actually stopping, in this case, production of a cellular gene that would otherwise make this protein. And it might seem it's very inefficient for the cell to make a messenger RNA and then degrade the messenger RNA, but that probably makes more sense than continuing to make a protein that the cell wouldn't otherwise need. So this miRNA system allows for an additional level of protection or an additional level of control of cellular gene expression. In any event, what happens here is that the translation of the messenger RNA is stopped. Now, I should mention the fact that this processing that happens does not have to cut the RNA. It can also involve a simple binding of that miRNA or siRNA sequence to the messenger RNA and stop translation by the formation of that duplex alone. Well, the last place that I'll talk about here where the RNAs get involved is, of course, in the carrying of the genetic information to the ribosome via the genetic code. The genetic code, as I've discussed in another presentation, is a 3-base sequence that determines ultimately how proteins are made. Each 3-base sequence, called a codon in a messenger RNA, specifies the incorporation of an amino acid into a growing polypeptide chain. Now, you can see the actual genetic code that's up on the screen here. And it's beautiful in its simplicity and also beautiful in what functions it performs. As I've noted before, there is a sequence called a start codon that is involved in starting the synthesis of every protein that's made in the cell. That start codon is known as AUG, and it codes for a methionine, as you can see in the first column near the bottom. There are also 3 codons called stop codons that tell the ribosome this is the place to stop incorporating amino acids into a growing peptide chain. They're known as UAA, UAG, and UGA, as you can see on the screen. Now, the genetic code really is what we all rely on to exist. Without the genetic code and the proper use of the genetic code, life as we know it would not be possible. The genetic code is universal. That's one thing that's very interesting about it, and it says a lot about our relationships to every cell on the face of the earth, because every cell on the face of the earth is using the same genetic code. That turns out to be not only just an interesting fact, but also important when we think about how we want to use the genetic code to make products in biotechnology. So, for example, if I were interested in producing, let's say, human growth hormone, and I wanted to take a bacterial cell and have that bacterial cell make that human growth hormone, I could actually do it surprisingly easily. I could take the coding sequence for the human growth hormone and remove all the introns and all the things that would be happen to it in a eukaryotic cell so that I have all the spliced version of that gene, and put that gene into a bacterial cell under the control of a promoter. If I take that gene and do that, the bacterial cell will transcribe through that coding region for the human growth hormone, and then the ribosomes will grab it and start to translate it. And since the genetic code is the same for a bacterium as it is for my cells, the bacterium will actually start producing the human growth hormone protein. That's a remarkable thing that we can do as a result of this common genetic code across organisms. Now, another consideration with the genetic code, and it's one that isn't commonly discussed, is the genetic code relies completely on accuracy. We saw during the modules on DNA replication the extreme lengths that cells went to in terms of proofreading and correcting errors and correcting damage in the DNA, because we knew that the integrity of the DNA was critical. And the reason it comes down to be critical is because the proteins that are made from it have to be accurately made, and if they're not accurately made, there can be severe consequences. But it's not just DNA replication that was important. The process of splicing was also important. We remember that RNA polymerase copied the DNA, and in eukaryotic cells, those little intervening pieces had to be removed. And the removal of those pieces has to be precise, because if they're one nucleotide, one direction, the other, the wrong way, then the entire message is ruined, and the protein that would be made from it is ruined. The last thing that has to happen relative to the actual sequences is the processing of these individual RNAs. The processing to make them so that they're translatable, to modify the RNAs as necessary to improve their function or to facilitate their function is essential. So without these things going on, the genetic code would have no meaning. Now, it's interesting that the translation process is not as accurate as DNA replication. For a cell, let's say a bacterial cell makes an error in replication about one time in every 10 million bases. That's a pretty remarkable set of accuracy, especially when you consider it's working at 1,000 nucleotides a second. That's a lot faster than any typist, and I don't know any typist who can type that accurately. That's remarkable. But translation does not need to be as accurate as DNA replication. Now, that's the sort of flip side of this thing. We need the accuracy to have the translation proceed properly, but the translation can actually have a few errors. If the translation has errors, the proteins that will be made will not function. They will be destroyed. And as long as those errors are relatively infrequent, most of the protein that's made will be functional. Now, if we have an error in the DNA, that has very different consequences, because an error in the DNA, well, that ruins everything all the way down the line. And as DNA is being passed from one generation to the next, once you've gotten an error, then that error carries forward. So errors in DNA replication have major implications for the genetic code and the translation of proteins, but errors during the process of translation can be tolerated to some extent. So the ultimate function of the genetic code and the ultimate value of the genetic code is in the accuracy of all these processes, and the last part of it is putting the right amino acid onto a transfer RNA. Now, the transfer RNA, of course, is the molecule that carries the amino acid to the ribosome. And in the ribosome, the anticodon loop of the transfer RNA pairs with the codon. That can be fairly accurately established. However, if the transfer RNA has brought the wrong amino acid in, even though the codon-anticodon pairing is good, the wrong amino acids will be incorporated into the protein. So it's very important then that the amino acid at the 3' end of the transfer RNA be the one that corresponds to the anticodon at the anticodon loop. This sequence that's there is actually read by an enzyme called the amino acyl tRNA synthetase, and it does what you see on the screen here. It reads the anticodon and puts the right amino acid onto the 3' end. Amino acyl tRNA synthetases have to get it right. If they don't get it right, then everything else is out the door. So the ultimate integrity of the genetic code is residing in the catalytic activity of these enzymes. So how do they perform what they do? They actually look at both ends of a transfer RNA. That means they have to be able to span from the bottom where the anticodon is up to the top where the amino acid gets put on. So I'll tell you a secret here. Most amino acyl tRNA synthetases don't span the entire distance from the anticodon up to the place where the amino acid is attached. They're not physically big enough. And this was realized when people began to analyze the three-dimensional structure of a transfer RNA. And when they did that, they discovered that the transfer RNAs that we typically write in a two-dimensional form, as you see here as a flat structure, really isn't a flat structure, but in fact is bent about in the middle. And that brings the end that gets the amino acid onto it much closer to the anticodon, so that the span of an amino acyl tRNA synthetase can actually cover both of those areas at once and have the proper amino acid put onto the transfer RNA. So when the proper amino acid is linked, as we've seen here, then the transfer RNA is ready to go to the ribosome and be translated. Now, this is pretty important to get right. So cells invest a fair amount of accuracy and a fair amount of energy into the synthetases that do this. There's one synthetase made for each amino acid. That is, it's specific. There are 20 synthetases that are used to put amino acids onto tRNAs, one for each of the amino acids that goes into a protein. Now, those synthetases have to be able to read slightly different anticodons at the end because the code is redundant, meaning that some amino acids are specified by more than one set of codons. But those synthetases have built that into them, and as a result are able to put the proper amino acid onto the transfer RNAs. Now, there are two different types of synthetases that cells have. One is called a type 1 synthetase, and these synthetases put the amino acid not actually on the 3' end. It's on the end that has the 3' end, but they scooter over one and put it on the 2' hydroxyl. They kind of cheat. The type 2 hydroxyls actually do what I've always said all along, is they put it on the 3' end. That is, they put it on the end and on the 3' hydroxyl. In either case, it's going on to the same piece of DNA, and it's going on in the orientation you see. It just depends which portion of the ribose sugar that it's actually going on to. Now, an interesting thing about the amino acyl tRNA synthetases is, like the DNA polymerases that we saw, amino acyl tRNA synthetases also do proofreading. So as I said, translation doesn't have to be 100% accurate, but it's important enough that the amino acyl tRNA synthetases are checking after they've put the amino acid onto the tRNA. They're checking to see that they've got the right amino acid corresponding to the anticodon. This helps improve the accuracy of this process I've just described to you. Well, I've talked in this lecture a lot about RNA. I've talked about how RNAs are processed. I've talked about how RNAs can perform functions including their own regulation and catalysis upon other RNAs. In the very end, we've seen how the RNAs have to be carefully processed in order to be translated. And I hope what I've left with you in this discussion is the importance not only of the function of RNA, but the things that RNA is doing in making proteins.